Thank the Human Frontier Science Program for awarding us this grant, because it's given us all an immense amount of pleasure. We've got 4 groups across the world, and it's been a very productive 2 years so far, and I'd like to share some of our results with you. So our previous speaker spoke about symmetry and breaking symmetry, so we're going to go from different asymmetrical peptides to asymmetrical flowers. So, the botanists in the audience might have heard about stylar polymorphisms, and heterostyly is one of the most well-known stylar polymorphisms, where you get a long morph and a short morph. What is more unusual is when you get the style deflecting either to the left, which we call a left morph, or a right morph, and that's called enantiostyly, and that's breaking symmetry. So there are many examples where we get both left and right flowers on the same plant. So early on today, you saw a picture of a tomato flower being buzz-pollinated. And if you go home and look at your tomato plants, you'll see that some of them have a left morph and some of them the right morph, but it's always on the same plant. And we call that monomorphic enantiostyly. And this is a sister genus to the Wachendorfia that we've been working on. And if you look carefully at this flower, you can see the blue arrows are showing a left morph, and the red arrow is showing a right morph. And so you see both these types of flowers on the same inflorescence. What's very unusual is what we call dimorphic enantiostyly, where a plant is either entirely left-handed or entirely right-handed. And when we wrote the grant, there were 3 examples we were going to use. One was called Heteranthera multiflora. It grows in Missouri. They're the Hemideraceae family. There are 4 Wachendorfia species, and also a sister genus called Barberetta, aurea. And then there's a third group called Cyanella alba. So the Heteranthera multiflora, one of the postdocs spent 10 days searching through Missouri last year, and she couldn't find any R morphs. So the Heteranthera is a bit on the back burner. The Cyanella alba, we've just submitted a paper showing that it, in fact, is not genetically determined. I'm not going to talk about that, but what I am going to talk about is the Wachendorfia, because there we have very strong evidence that it is indeed a genetically determined dimorphic enantiostylous species. So I'll be talking about 4 species of Wachendorfia, and then a sister genus called Barberetta, aurea. What are the questions we're asking? So we start off from the ecological and evolutionary approach, which is what's the ecological importance of dimorphic enantiostyly, and I'll share some of those results with you briefly, and then we'll go on to how is symmetry actually broken, and how we've used genetics and cell and developmental biology to answer that. Okay, so the first question is, if we go and count Wachendorfia paniculata, I'm very lucky, they actually grow behind my house in Cape Town, which makes for a very easy experimental system. And if you go and walk down the fire break and you count them when they come up, there's exactly a 1-to-1 ratio of left morphs versus a right morph. So this is Cape Town. You can see Cape Town's down in the bottom left-hand corner. Barberetta aurea grows in KwaZulu-Natal. There's showed the picture of it on the stream here. And if you count those also have a 1-to-1 ratio. So how is this maintained? So you can imagine it might be well maintained if there was a genetic incompatibility system by which a left morph wouldn't form a functional cross with another left morph, or an R morph couldn't form a functional cross with another R morph, and that would always promote left versus right and right versus left. So one of the first things we did was to check for that incompatibility, and our conclusion is that there are no genetic barriers to intramorph pollination. So on the left-hand side, you can see the data for paniculata. This is, once again, for the biochemists and the physicists amongst here. This involves taking pollen from an anther, from an intramorph cross. So you would take a pollen from a left anther, and you would put that onto the style of a left morph plant. We call that an intramorph. If you go from a left anther to a right morph plant, that would be intermorph. Autonomous self means that you bag the flower and you don't allow anything to pollinate it. And manual selfing is you manually go and take the pollen from the one anther and put it onto that style. So you can see that the autonomous selfing on the left for both Wachendorfia and for Barberetta aurea is extremely low. Okay, so the plants on their own don't set seed unless some intervention takes place. You'll see that for Barberetta aurea on the right-hand side, there's not much difference in terms of seed set between manual selfing, intramorph, and intermorph. For Wachendorfia paniculata, there was a bit of weak self-incompatibility, you can see with the manual self-sorting, but no difference between the intermorph and the intramorph. So the question then is, how do we maintain these 1-to-1 ratios that we actually see out in biology? Well, the answer is we should maybe think about this enantiostylus more as an adaptive system for wing pollination. So if you look at a Barberetta aurea flower, you can see it on the top right there in B. It's got little markings on the upper petals, and those serve to actually guide the insect to come in and take the nectar out of it. And I'm going to show you a video here that was made by Steve Johnson, which shows you very elegantly how a syrphid fly comes down and lands on the flower in a very precise way that the wings touch the style on the one side and the stamens on the other side. So this Let's get away from that horrible noise. Sorry about that. Okay, so let's move on to the cell and developmental biology and the genetics aspect of this. And so the question is, how do you break symmetry in plants? So I guess with all of research, you go and you look at the literature, and there's a bit of a literature in Arabidopsis and Arabidopsis mutants where roots of an Arabidopsis usually go down straight, and there's some mutants where the roots go to the left, and the other mutants where the roots go to the right. So if we look at this, if you've got a normal root in the middle there, you'll see that you get things called cortical microtubules. They're a bit like the spokes around a barrel. And if they're in the horizontal plane like that, you get perpendicular growth going up like that. Okay? And you'll see the cell files on the external part of the root are in a perpendicular direction compared to these cortical microtubules. Now, what you find in the lefty mutants is that over here is that the cortical microtubules are at a negative angle, and the growth then occurs at 90 degrees to that, growth occurs at 90 degrees to those microtubules. And then for the right-handed growth, we get the opposite. And you can see that quite clearly over here. Over here, these are the SEM data in that figure D, and you can see that the cell files are on a negative angle. So one of the first things we did is that we went and thought, well, okay, let's go and look at our styles and our stamens under a scanning electron microscope, and let's see whether they're orientated and are they twisted, either to the left or to the right. Okay, so this is what we did, is you take your style and your stamen, and you can put it on these grids. And then just to remind you, is that showing over here? No. Okay, so on the top left, you've got a left morph flower, and there we would predict that the cell files should be a positive angle if we've got left-handed growth. And for an R morph flower, our cell files should be at a negative angle if we've got right-handed growth. And so what we did is we took SEM images, and we measured the angle of the cell file relative to the longitudinal growth section of the stamens and the styles. And this is just to show you what the images look like. So this is at the base. You've got a left morph and a right morph. And then if we actually plot these angles, on the top right there, you can see that at the base, the right morph, where we would expect to get negative angle, in fact does have a negative angle, specifically at the base, whereas the left morph, we expect to have a positive angle if it agrees with the Arabidopsis mutant type thing, and in terms of growth to the left, and we in fact do get a positive angle. But that twist seems to be happening at the base, because we can't see any other permanent twisting at any other section along either the style or the filament. If we look at the filament, we get the exact opposite, which is what you'd expect, because if your style is going to your right, then your filament's going to the left. And here, our right filament has got a positive angle, and the left filament's got a negative angle. So that all looked quite elegant, and we've been doing a whole lot of work to try and see whether we could actually stain the cortical microtubules, and that's quite a complicated thing to do if you haven't got a GFP fusion to a microtubule protein, to tubulin. So we thought, well, let's try and do a video image and see what happens while these stamens are growing. So this is a bud. We've taken off the petals, and we've stuck it into some agar, and we're watching it grow over time. So it's a right-handed flower. It's a right morph, and you can see that our style there goes into a right morph direction. And you can see that as it's actually growing, it actually does a bit of a snake dance. So we call this the snake dance video. The other important point to note on this is that the symmetrical anthers are on the top, so they're on the adaxial surface of the stamens. Now, the student, Caroline Robertson, who did this video, was writing up her master's, and so I thought I would go and make another video. And when I did that, I put the flower upside down by mistake, not realizing it. And we got a super surprise. So, if you've got a normal flower, we've got over here, Okay, so on the right-hand side, there's our normal flower, and I've put on the right-hand side, I've got the picture of the flower, and you can see that little blue arrow is pointing to the style on the right. Now, if you turn that flower upside down, okay, and if the right was determined exactly by the flower, our stamen should actually go to the left. Okay, but what we find here with these inverted flowers, and if you look at the inverted style, you see it's going to start going to the left, and then it changes its mind, and it goes completely right. So you can see it's starting to go left, and then it says, hang on. And in fact, it goes entirely to the right. So my mistake has turned out to be really, really interesting. So we've added a gravitational response to how the style is being deflected in addition to these cortical microtubules. So then to try and check this, we thought, okay, hang on, we've taken these buds and we've put them into MS agar. Is that a real experiment? Let's turn these flowers upside down and to see how they behave. So we got a good old retort stand, and we put the inflorescence upside down and compared that to an inflorescence that was upright. And so on the extreme right-hand side, you can see the control after 2 days. The little red triangles are showing you where the opposing stamen is, and the blue arrows are showing you where the style is. So this was an L-morph inflorescence. And then in that column labeled A, you can see the blue arrows, instead of being, it should be upside down, so it should be on the other side, but in fact, it stayed where it should be. Okay, so that seems to be holding. So the next thing we thought about was, what happens if we rotate the flowers? Okay, so if we put them upside down, they go in the one direction. So if we keep rotating them, well, you might predict that the style would then go straight because the gravitational field is changing all the time. So we took our buds and put them into Eppendorf with our MS agar, and we put them onto this little whirly mixer, and they went round and round. This is the wild-type flower control that wasn't exposed to this torture. Here are the 3 that were continually turned, and you can see these little blue arrows are pointing to the fact that the wild-type flower, the L-morph, is completely on the left-hand side, whereas these ones that were rotated continually for 24 hours, the style went dead straight. Okay, so our model we're working with at the moment is that there might be an initial setting up of a right-handed and a left-handed microtubule helix. Those then are converted into a left or a right-handed cell file twist, as we've seen in the SEM data, but then there's a gravitopic response that results in a stronger cell elongation on the basal side of the styles. And that's how we get an L-morph and an R-morph flower. So you can ask, well, what is the genomic difference between L and R plants? So this is a view of Cape Town, and our study site is over there, so it's very beautiful. And what we did is that we went along at the start, before we wrote the grant, is we collected 110 left-handed plants, 110 right-handed plants, pulled the DNA from those plants, and sequenced the left and the right, and asked, what's the difference between them? And what we found is a whole lot of contigs that were only present in the right-handed plant, and there was about 1 megabase worth of multiple contigs. So let's look at characterizing this, what we call the E locus. So we set ourselves a number of questions. First of all, is it contiguous? Because this was short read sequencing, so we had multiple contigs. Is this region conserved across the 4 Wachendorfia species and Barberetta aurea, which is a sister genus? And are genes in the hemizygous region expressed in developing styles and stamens? So we did long-read sequencing, and we assembled the genomes of Paniculata and Barberetta aurea, and we could confirm that there's a 400 kilobase region in Wachendorfia Paniculata that is contiguous and is only present in the R-morph flowers. We then collected 50 left-handed and 50 right-handed buds from either the other 3 Wachendorfia species. And this is a picture of Mbali, who was doing an honors project on that, and this is where we collected the Wachendorfia multiflora buds from at Langebaan. Okay, this is just to take you through this fairly quickly. These are the 3 other sister species to Wachendorfia paniculata. And by doing a comparison of our short read sequencing, we could narrow the 400 kilobase region right down to 80 kilobases that was conserved across all 4. Now we can ask the question, we've narrowed this down, are there any transcripts in this region that are expressed in the developing styles and stamens? So we started off looking at the styles of Wachendorfia paniculata. I'm not sure why we just concentrated on the styles in the first place, but we did. And to our surprise, if you look at this hemizygous region, we had identified this gene YACA-R that's present in the hemizygous region. It's involved in auxin biosynthesis, and so we thought, that's a fantastic candidate. When we looked at expression with an RNA-seq analysis, there was 0 YACA-R expression. Instead, there was a non-coding RNA that was expressed in the right morph flowers. When we looked at this non-coding RNA, and we did a blast sequence, it's similar to a PRI-MER156. It's got high similarity to the Arabidopsis PRI-MER156. And if it's processed, you'd expect to get short MER156 and the complementary MER156. So we went to the sister species, Wachendorfia multiflora, and asked, do we see the full-length PRI-MER156 in it, and do we see the processed versions? And the answer was yes. If you look at the styles, we get the full length in the right, and we see both the MER156 and the MER156 star in the Wachendorfia multiflora. So it really does seem to be real. What about YACA-R? As I said, it's involved in auxin biosynthesis, and the question is, where is it expressed? So we looked at both stamens and styles in Wachendorfia multiflora, and we see very strong expression only in the right-handed styles of Wachendorfia multiflora, and we repeated the experiments on paniculata, and we see strong expression in the right-handed styles. So what's the functional importance of this? We've got a MER156, we've got a YACA-R. Now, the challenge of working on a non-model plant is that you can't go and do the standard types of genetic tricks that you can do with Arabidopsis, and so you've got to be a bit creative. So one thing we thought we could do is try and overexpress this PRI-MER156 in Arabidopsis and see if we get a MER156 type phenotype, and also look for naturally occurring mutants. So to just quickly show you, this is what the Arabidopsis overexpressor looks like. On the left-hand side is wild type. On the right-hand side, you get this retention of the juvenile phase, so multiple leaves. And if we express Barberetta aurea overexpression in Arabidopsis, we get exactly the same phenotype. So we really do think this is a proper MER156. What it's doing in Wachendorfia remains to be seen. So then we had this crazy idea of let's go and look for naturally occurring mutants. So on the left-hand side, you've got the L morph. The right-hand side, you've got the R morph. So you can imagine if we had a deletion in the MER156, what would happen is that opposing anther, the opposing stamen would, the right-hand side, if we've got a deletion of our style, the pink one, that'll go across to the left, and we'll get a left homostyle. If we've got a deletion in YACA-R, then the stamen won't develop. It'll develop like an L-morph stamen, and so we'll get a right homostyle. So we were actually going past the nuclear power station, and we went, let's go and look for some mutants, and we found some. So, we did a whole lot of mutant hunting. So this one is not from the nuclear power station. It's from Rondebosch Common, which is quite close to UCT. And we found what are left homostyle mutants. And we would predict from the previous one that they might have a deletion in MER156. We found at a place called Riedlinghuis a bunch of R homostyles, which would have a prediction with a deletion in YACA-R. So we've sent these mutants off for sequencing. And so if you look at our left homostyle, which is in red, we predict a deletion in MER156. And if you follow this red curve over here, YACA-R is present, but when we get to the right-hand side with the red line, you can see that MER156 is completely absent. And if you look at our right homostyles, and those are the blue and the green lines, where we predicted you'd have a deletion in YACA-R, if we map those reads, indeed, we do have a deletion in YACA-R, but the MER156 is present. So this is our best evidence at the moment we've got, that these are really functionally important for determining the left and right morphs of Wachendorfia and Barberetta. So that's our model. Yaka R is important for development of the stamen, MER156 for deflecting the style to the right. How that is linked to the gravitropic response and to the cortical microtubules, that's our next challenge, to make that link. And then just to finish off, it's been a huge group and it's been a lot of fun. We've got University of Potsdam, Toronto, Wageningen, and then a bunch of South Africans. And we're very lucky because these plants grow on our doorstep. So thank you.
Thank you. Because of the time, just one question.
Hi. Yes, thank you for this amazing study and the excellent talk. Yeah. So I have just one question about okay, so do you think that these selection for these morphs are basically because of the fertilization that they have, like the reproduction of the plants, so to make them more self-reproducible, or to have the pollinators to be more generalistic, to attract broad range of pollinators to specialized pollinators for these plants?
So I'm not sure if I understood the whole question,
Okay.
but just in terms of pollinators, I think you actually need quite a precise pollinator to maintain those 1-to-1 morph ratios.
Yes.
Because the plants, there's no barriers to intramorph pollination. So if you get a bee coming in,
Yes.
and the bee turns upside down and goes in at multiple angles, then you're going to get a lot of intramorph pollination happening.
Yes.
So I think the plants evolved to specialist pollinators where you've got very precise deposition of the pollen on the wings. So if you visited an L-morph flower and if you then go to an R-morph flower, you'll very precisely put the pollen onto the style of the opposite morph. So we
Yes.
see a breakdown in that 1-to-1 ratio, which I haven't spoken about.
Yeah.
And we're interested in what makes that breakdown.
Yes, so I got that. And so what I'm asking is that, so this specificity that has been evolved in the plants is basically because of making them to attract only selected pollinators, to facilitate selected pollinators rather than broad range of pollinators to, yeah.
So I think one of the things that always surprises me at home is we don't have that many insects. So I come to North America, I get bitten to smithereens because you have so many insects and biting things. So I think in this sort of very nutrient-poor environment of the Cape Fynbos, there are not that many insects, so you've got to specialize
Yes.
to get efficient pollen transfer. And I think that might have is what's driven it.
Yes. So, all these species are actually self-pollinated or pollinator-dependent ones for reproduction. So, these species that you have worked on, are these all self-pollinatable species, or?
Self-pollination is possible, but if you just look at autonomous selfing, so if you
Yes.
hand self it,
Okay.
you'll get it'll set seed.
So
Okay? But if
they.
you don't do that hand pollination, the style and anthers the autonomous selfing is very, very low.
Yes, so they cannot actually in the wild can self-reproduce, right? So they are dependent on the pollinators.
Yeah, they do have bulbils. We can maybe chat at tea, but they have
Yes.
bulbils, so they can also vegetatively propagate by extra bulbs.
Okay.
So we're interested in
Yeah.
when you get that breakdown in the 1-to-1, and we think it's when that syndrome breaks down. But let's chat at coffee.
Yes, we should share. Thanks.
Okay, thanks again.